All right, so good morning, everybody. Thank you all so much for joining us this morning. I'm really excited to introduce our presenter this morning, but before I get too far ahead of myself, a little bit of housekeeping, which is to just say that we do have our speaker for next month planned. And so if you've ever wanted to know more about how bat houses and artificial roosts are used in bat research specifically, then feel free to sign up for next month's talk. Our speaker is going to be newly minted Dr. Reed Crawford from the University of Illinois in Champaign-Urbana. So if you've ever been curious about how bat houses are used in research, please feel free to sign up for next month's talk. Otherwise, we can sit back and enjoy today's speaker, which is another topic that I absolutely love. Even though I do not personally use museums in my own research, I have always been an avid fan of museums, museum collections, and visiting, and have helped in a number of natural history collections myself. So the fact that Connor Burgin agreed to be our speaker this month, I am very, very happy about because he is um, definitely a museum fanatic and has been working in museums in mammal collections. Like what you've been interested in museums since you were a kid, practically. Yeah, so he's been working in museums for a number of years, all throughout his PhD program, and is also was named the American Society of Mammalogists Fellow in 2023 for his work in museums and has helped put together the Mammal Diversity Database. So he does a lot. And so without further ado, I am very excited to introduce our speaker this morning, Connor Burgin, and he will be presenting why and how are museums used in bat research. So take it away.
Awesome. Thank you, Dana. And just making sure my audio does sound good, right? I'm assuming that's a yes.
Yes, sorry.
Okay. I'm Connor Burgeon. I'm currently a PhD student at the University of New Mexico, but I'm originally from Boise, Idaho, which love it there, but New Mexico's way cooler at this point. And I'll be talking about why and how museums are used in bat research today. However, I will also be talking about mammals more broadly. Most of my research is not just in bats, but I've tried to cater this talk more towards a general audience and about bats. And a quick disclaimer to anyone who may be sensitive to dead animals. There will be pictures of museum specimens and deceased animals in this talk. Also, this is a picture of a bat that we got a little bit ago doing some field work in New Mexico. Midas the cenotes. He's very cute. So, the general consensus by the public about museums is that most people really just know it as public exhibitions. This photo is from the Smithsonian. It's a very charismatic one. Most people would know there's a giant elephant that you look at every time you go into the Smithsonian National Museum of Natural History. But some people may also know that there are archives associated with museum collections. This is a good picture of some fluid specimens at the Museum of Southwestern Biology here in New Mexico. And others may also know it from its education outreach. These are specimens that I had pulled out for teaching a mammalogy class last semester. But the natural history museums have a lot more uses than just what the general public knows, um, such as being archives of historical information, centers for collaborative research, something I'll be focusing really heavily in this talk, and repositories of untapped knowledge, as well as resources for public education. So, those three general facets that the public would associate with museums do hold true, but there's a lot more complexity to it, especially with the use of the specimens that are collected by the museum researchers. So, oops, up, down, there we go. So, we use specimens for a lot more than just looking at. Sorry, my mouth's very dry and I don't have water. Sorry. There we go. Um, we use a lot more than we use specimens a lot more for than just public outreach. We also use them as a record of an organism at a place at a time, which I think is the most central aspect of museum collections. You're collecting a species, and that species is a voucher of that organism at the specific place at the specific time it was captured. Then it can also then be used for applied research that we'll talk about later, such as systematic work or conservation related research. And then it can also be used for documenting biodiversity more generally and the distribution of organisms, which is why we want to collect over a broad area and over an expansive time so that we can track those changes. So some of the ways in which species, bats in general, are collected are using large nets called mist nets or harp traps. Um, and because I work with mammals more generally, I wanted to include a little Sherman trap with this little silky pocket mouse. And again, the bat that we collected last summer. And when we're collecting specimens, at least with most museum curators, try to utilize as much of the specimen as possible when possible. So here at the Museum of Southwestern Biology, we use a holistic collecting mindset in which we collect more than just the skin and the skeleton of the animal. Of course, we do collect that, or the whole animal in ethanol, but we also include the associated measurements, standard measurements like head body length, hind foot, ear, and in bats, we specifically will record forearm length and the length of the tragus, which is the little notch on the inside of the ear, which are important for species identification. Um, and going back to each of these specimens being a record of a species at a place and a time, we also have the locality information as specific as possible and the date that it was captured. Now, what this extends into with the holistic collecting mindset is including preserved tissues and blood, which, at least here at the Museum of Southwestern Biology, we try to put in liquid nitrogen immediately so that it is automatically frozen and preserved in the state that it was when the animal was sacrificed. This is for to make downstream application of those tissues a little easier, such as genetics or looking for RNA in those tissues for other applied physiological research, and so on. And we'll also collect blood, which can be used for screening for viruses and bacteria that may be found in that host organism. Additionally, we will collect endo and ectoparasites, which aren't always collected, but are collected as often as possible. This photo comes from some of the researchers and collaborators of the Museum of Southwestern Biology and Kansas State University when they went to Panama last year. And this bat is a vampire bat, as you can see. Very cute little guy. But additionally, we'll collect more tissues. We at least collect seven here. But I know that there's also different research tactics when there's less funding available, or if is an inability to collect broadly, where sometimes we have to collect specific tissues from specific organisms, and that is also an important way in which we collect specimens. But collecting holistically is often the best way to utilize the specimen as best as possible, which is important to us as museum people. Like we got into zoology and biology because we love these animals, so we want to use them as best we can. So why should we keep collecting? I think the most basal reason is we're still trying to document the world's biodiversity. There are new species being described yearly in all groups of life, including bats. And if we don't know what we have, how can we conserve it and how can we study it? And a more broad reason for collecting more is to have longer time series. So we want to collect from the same place multiple specimens over long periods of time so that when we do genetic analyses, such as this one on the right here, which is not of bats, but of another group that I work on called lemmings. um, we have comparison across multiple time periods from the same locality, which allows for repeatability in our research. Additionally, we have unknown applications for the future. This is really exemplified by genetic data in the modern day. So, if we look back at historical museum collections, there were researchers collecting these specimens and doing morphology work, which is looking at the form and function of the structures on the organisms. But they were completely unaware that genetics may be used on these specimens 100, 200 years down the line. And we are able to collect DNA from from specimens collected as far back as 100, 150 years, and maybe even older as technology progresses even more. And I know some pretty old ones, but it's a good exemplification of why we should collect these specimens for future use that we may not know at the time of collection. So I wanted to highlight the Museum of Southwestern Biology. This is where I personally am working, and I focus on mammals more broadly. So this museum holds a special place to me, not just because I work here, but because it has the second largest collection of mammals on the planet, second to the Smithsonian at this point, which is why I love it here. And the other reason why I like it, which is kind of specific, but the mammal division, its emblem includes the spotted bat, which is currently my favorite species of mammal, besides the three-banded armadillo, and also my favorite bat. And additionally, there's some symbolism to this because the museum holds a specific specimen of bat, which I was going to include a picture, but I did not have time to go get a picture. But we have a specific spotted bat specimen that is mummified from a cave that was identified to as being 10,000 years old, which is really interesting to have in a museum like this, when we have modern specimens of spotted bats just sitting in the museum as well. Um, I think the Museum of Southwestern Biology exemplifies some of the most important aspects of museum curation, and as it relates to research in general, as most museums do, but I work here, so. But we have dry specimen archives and ethanol specimen archives, as well as a division of genomic resources, which are these large nitrogen vats that hold thousands of small little tubes with tissues in it. And the goal is to preserve those tissues as best as we can for as long as we can, and use them to extract genetic material or whatever, or be used for whatever research it needs to be used for. And an important aspect of this is collaboration with external researchers, because what's the point of a museum collection if you're not using it? So we try to loan out those samples as best we can to other institutions. But we also do in-house research, and we have a prep lab space for a lot of that, plus external lab spaces for genetic work. I didn't have a picture of our lab space, but that door in the background behind this lion, that's where we work. But the lion's pretty cool too. And additionally, we also do a lot of public outreach. This picture is two of our grad students in the museum, showing some of our specimens to a group of high school students. And as you can see, there's one person that is very enthusiastic about it, and I love that because I would also be that kid in high school if I didn't have this resource available to me in Idaho, unfortunately. So it's really nice to be able to see that there are students that get to come and utilize these collections and learn about them. I recently had some students come through and I taught them a bunch about bats specifically, and I got a picture, but I looked really bad in it, so I did not include it. But I wanted to focus a bit on the tissue storage. Here's another picture of the Fields Museum tissue storage that they have. And the Museum of Southwestern Biology specifically has worked with a number of international organizations and worked on specific grants, including this one called Picante, which is for pathogen discovery, specifically building pathogen informatics centers. And part of that is collaborating on filling loan requests across the globe as best we can and creating in-country resources in non-US countries so that they don't have to rely on the US at any point in the future. So, wrapping this back around to bats a little bit better, this is a map from 2018, so it's not the most up to date of bat species distribution richness. Sorry. And I think, honestly, just aside, this is a really interesting map because it shows that there's a huge density of species in South America, which I'm not sure if South America fully is going to be the place that has the most bat species, or if it's just a relic of the fact that a lot of new bat species have been described from South America lately, whereas other regions of the world still have a large amount of undiscovered diversity in bats. Um, But museums are important for a lot of aspects of bat biology. One of those is bat conservation, specifically with disease detection and tracking. White nose syndrome and is probably the most commonly talked about bat-related illness, and it's been ravaging North American bat populations ever since it was introduced into the populations in the East, and it's been moving west across the continent. Um, Museum specimens, I don't think have been utilized to my knowledge, specifically for white nose syndrome detection, but the specimens can be vouchered and tracked over time. And with further techniques developed, we might be able to track white nose syndrome into the past using the museum specimens that are already collected from these localities if there's enough sampling. So this is highlighting the fact that we need collections over times across all species in case these major diseases may be affecting their population. And we could go back into the museum archives and work to um, find a source of this disease that may allow us to deal with the population crashes better. Additionally, museum specimens allow us to track population structure over time and space in various bat species and just all species in general. But some of these examples, this is the Hawaiian hoary bat, Lasius simotus, known for being the only terrestrial mammal species or fully terrestrial mammal. Well, I guess it flies as volant, but it's the only species of mammal that lives in the terrestrial part of the island permanently. Um, and it was recently split off into its own species, separate from the North American or the hoary bat in general, which was now split into three species. But using museum samples and newly collected samples, there have been recent studies looking at the population structure of Hawaiian hoary bats across the islands that has allowed us to, or allowed researchers to investigate the effects of various aspects of human existence on these bats and their conservation in general. Additionally, we can look at population structure across large distributions. Um, I think I can't remember what this species was, but it's a European bat species where they were looking at the structure of different populations and looking at whether there is genetic material being moved between these populations. So we can look at how they're structured and distributed across the landscape. And that allows us to better influence conservation litigation for these groups or these species and populations so that we're not mixing populations that may be best kept apart, or we're allowing for genetic material to be mixed between these populations. This often gets talked about with large mammal translocations a lot more than it does bats. But I think that it may apply to bats in the future as we develop more conservation actions for certain bat species. Additionally, museums have played a central role in disease detection and prevention, or zoonotic disease detection and prevention, as it relates to human health. COVID-19 is the poster child for this at this point, because SARS-CoV-2, yeah, seems to have originated in bats based on current knowledge, but that was debated heavily amongst the community. This paper's from 2020, and they were trying to identify an intermediate host from the bat to the person, or whether it went directly from bats to people. And some of the best ways we can look at this is by collecting specimens, vouchering those specimens, and screening for the virus. The Museum of Southwestern Biology, in particular, had a grant specifically for looking for coronaviruses or related coronaviruses by screening for animals in Asia. So we had a large collection of specimens from Mongolia that were collected over the 2000s, and we were able to screen the tissues for coronaviruses, discovering novel coronaviruses and viruses similar to the COVID-19 coronavirus. So, additionally, this is a non-bat related example, but I had to mention it because it's also a major story from for advocating for the utility of museums through the Museum of Southwestern Biology. And it's one that we talk about on every tour we give at the Museum of Southwestern Biology. But the Sinombre virus, sorry, one second. Still left something on that is making a sound. There we go. Okay. So the Sinombre virus is a type of virus that had an outbreak in 1993 in New Mexico. And at first, it was uncertain where this virus came from, but it caused hemorrhagic fever in people, and it had a very high mortality rate. So to identify the host, the researchers at the time, Terry Yates at the Museum of Southwestern Biology, sequenced viral genomes from or screened for viruses in the various mammals in the collection. They found that the perpetrator of this zoonosis was Paramyscus maniculatus, which is now known as Paramyscus sonoriensis in the West. But it's the most common deer mouse in North America. So this is a widespread species with a very localized zoonotic spillover. And using these specimens, they were also able to identify that specimens dating back to the 70s had this virus in it. So this isn't a new virus by any means. But knowing this information allowed researchers to inform the public on how best to avoid getting this disease, which is mostly transmitted through fecal matter and urine. So, additionally, and this is where my research primarily lies. Museums can be used for systematic research, such as describing new species and looking at the biogeography of those species. Um, a recent example of a newly described species is Myotis nambayensis, which I think Nancy Simmons had a part to play in. She's in the audience. Hi, Nancy. And then hey, another example of this is actually last month's My Field for Dummies by Pedro Monaco. I really hope I spell it your name right, Pedro. In which is on biogeography in bats. And biogeography is a central part of systematics as how species are distributed and how they got there informs the taxonomy, and the taxonomy thus informs the biogeography by defining what a species is in that group. So I wanted to quickly go over some taxonomy stuff because I think this is an important aspect of the history of museum collections because a lot of the museum collections that were built historically were built for species discovery. Taxonomy is the study of naming organisms, and we name organisms based on a type specimen. And that type specimen, which I've indicated as little dots or stars, is the specimen or entity that permanently attaches a scientific name, that's permanently attached to a scientific name, that is acting as a reference to identify which taxonomic group a population or individual belongs to. So you compare the population in the wild to that type specimen, and you say, is this close enough to call it the same species? And taxonomy does change, however, as we use different data types to distinguish them. We also have a type locality, which is the specific geographic locality from which the type material or type specimen was originally from. So think of this circle as a distribution map, and each blue dot is a type specimen that is held in a museum. Um, while this big circle is a valid species, and all of these little circles are populations. But maybe another researcher comes in and splits those populations out into their own species. And the types localities of the oldest name then become the type locality of the full species. So there may be synonym names that get elevated and they get turned into their own species, or a new name has to be generated altogether. Um, in which case, a new species is described in the literature, and that holotype or type specimen is held in a museum. Furthermore, there can be more taxonomic divisions that result in a lump in some of those species. So maybe these two populations are found to be actually pretty closely related based on more recently published data, maybe genetic material. And then they are lumped together into a single species with two populations and multiple described names, but that a new name is now a synonym of another name. But because of the interconnected relationship between taxonomy and museums, it is really, really important to keep these specimens um, archived in the museums and tracked, which can sometimes be a lot more difficult than it sounds. An interesting example of I guess, interesting to me because I'm a taxonomy nomenclature nerd. Is this specific type specimen for Vespertilio aureus. It was supposedly from a island. Oh, I'm forgetting actually. There's there's two examples of this. One's from Singapore and one's from Samoa, and I'm actually forgetting which one is which. But it's either from Singapore or Samoa. Um, but it had bounced around from name to name for a really long time until recent um, advances in our ability to look at these specimens allowed us to identify what this species really is. So it was just considered Myotis aureus for a very long time until it was found to be a species of the genus Curavala. However, we were not able to identify which species it was, only that it was part of the genus, and it is now considered a nomen dubium, which is a term used in nomenclature specifically for names that cannot be fully identified. So taxonomy nomenclature gets really complicated, and that's why keeping track of these museum specimens is really important. So with so much data that is available for these specimens, especially if we have measurements and host parasite interactions that we're keeping up with. How do we share those? Well, I think the term biodiversity infrastructure is the best one to say for that because we have a lot of different connected resources on how to share that data. We have basal museum databases such as Arctos. There's also Symbiota and museum-specific databases such as the one that the National Museum of Natural History has in the Smithsonian. But there are also taxonomic databases like the Mammal Diversity Database and Bats of the World, which work together to keep track of the currently recognized bat species and their distributions and taxonomy. And those in turn will reflect on other databases such as the IUCN Red List, which is tracking conservation priorities for the species, well, all groups of life, but all bat species at this point, which is informed by the bats of the world and the MDD taxonomy. And then specimen-specific resources such as Morphosource, which is keeping track of morphological data such as measurements, 3D data, whatever it may be, from those vouchered museum samples. And then for genetic material, GenBank is often used, although its connections to the specimens in museums could be a lot better, as sometimes there's unvouchered molecular data that then does not have a source specimen to go back to, which can result in misidentifications, especially as taxonomy changes. And then we have other just specimen databases such as GBIF and VertNet. GBIF is pretty much all encompassing and also includes records of public records from iNaturalist, which is where people will take a photo of an animal or plant, put it into the database, and then others can identify it, or they can identify it. But a lot of my work focuses around the ASM's or American Society of Mammalogists Mammal Diversity Database. And I'll be talking a little bit about some of the global trends that I've noticed in bats and mammals, specifically associated with taxonomy and museums at this point. So a little bit of a shift. The Mammal Diversity Database specifically keeps track of all of the world's mammal species, their distribution, at least generally, and how their taxonomy is changing, as well as most recently the type material, which has been found through the help of Yellow Zylstra at the Hesperomys project, which is a nomenclatural database that has worked really hard to keep track of the world's or the mammals of the world's nomenclatural data. So their original citation, type specimen, type locality. Um, very, very yellow is great. I'm just going to keep saying that, just so you all know. Anyways, with this data, we're able to look at some global trends in mammal species diversity and new species discovery. This is a map that's a little bit outdated. It's from late last year, 2024, of new bat species descriptions since 2000, and it includes valid species in red that are currently recognized on the Mammal Diversity Database and bat names, as well as the blue dots specifically for synonymous names or subspecies. So they're not currently recognized as species. And there's some interesting trends we can see. There's a lot of names being described in specific localities, whereas there's various localities that have a lot less names being described. So Vietnam has a lot of names being described, Madagascar, and the Northern Andes in South America. However, we can see that there's other regions of the world that we might expect there to be a lot more species from, such as Southern Asia, Southeast Asia, that are currently have not had as much research done on the systematics of those species for various reasons. And there's also a lot of research being done in China at this point too, within country. which is also interesting to see on a global scale for mammals, but I did not include the full mammal map. But there are even new species being described in Europe. No bats in North America at this point, but new species are being described in Europe, which is pretty cool, especially Myotis. Then something that we kept track of between the Hesperomys project and the Mammal Diversity Database was the number of new species being described, or I shouldn't say it that way. The totality of all names that have been used for each mammal species, including bats. And this is a map over time of the names that have been described for bats, with the dark color being currently valid names and the light color being names that are now synonyms. And these lines represent the number of split species, so names that were originally a subspecies that are turned into a species through taxonomic revision, or lumped species, which are names that were a species that is now not a species. It is a synonym of something else. And there's some interesting trends associated with this, especially with this giant jump here. So this large jump in the late 1800s and early 1900s is associated with the British Museum, specifically with collections that were sent across the globe due to colonial practices that were then described as species by various authors in both North America and Europe, but especially at the British Museum, and particularly Oldfield Thomas, who is considered the most prolific mammalogist of all time. And he was a very significant museum curator in the history of both bats and mammals more broadly. So this very old museum houses a lot of the type specimens of these species. This is a map of where type specimens in mammals, specifically across all mammals, not just bats, are held. You'll see that there's a high density of museums in Europe and North America that have these type specimens, or a lot of them. I wish I could zoom in better. I needed to recreate this map, but the British Museum is right here, which has the most type specimens out of any individual museum. And this creates inequalities or disparities in where the type specimens of species are housed versus where those species live. Um, so we can see that there are a lot of species of mammal that are found in the tropics versus the northern latitudes, such as Europe. There's a lot lower density of mammal species in Europe versus the tropics. And include if we look at new mammal species as a whole, similar to that map I showed you earlier with specifically bats, looking at the type localities of new mammal species in the 2000s or 2000 and beyond, we see that most new names are being described from the tropics, with very few from the United States and Europe, while these regions still hold a lot of the type localities for many of those names. Although it is getting a lot better as more research is being done in the Global South countries by Global South researchers and being housed at the museums there. And we can see this really prominently in South America, where there are more institutions within those countries spread out that have type specimens in them now. And this map, I guess, shows this at a more staunch level. And we can see that okay, this is all names that have ever been applied to a mammal and where their type localities are by country. Um, this is showing that the United States has the most names described from it of any country, whereas countries in tropical regions are much lower on the list. Even Brazil, which has a very large area and encompasses most of the Amazon rainforest, um, but it has a lot less names than the United States does. Because Europe is fragmented to a bunch of small countries, but it would be interesting to see this at a continental scale, which I think is the next step. But it's showing that there's disparity in where type localities come from uh, versus where the type specimen of the species is housed. One last little tidbit, I guess, a little bit away from museum stuff. Um, we keeping track of taxonomy and these type specimens is important for many downstream applications in biological research. Um, all sorts of biological research rely on the fact that we have a defined species unit, including conservation work. So there's a direct train from collecting these museum samples to defining what these species are and their distribution, and to using them in conservation. Um, but this is a graph of the total number of mammal species as recognized on the IUCN Red List. This is not just bats, and bats, I think, have done a lot better job recently, especially with the connectivity between the IUCN's bat specialist group and bat names and the mammal diversity database to keep up with bat taxonomy on the IUCN. However, there's still about 25% of all mammals that are either data deficient on the IUCN, so they're recognized at least, but there's not enough data to assess them or not evaluated on the IUCN, for which there are 943 mammals that are currently not evaluated on the IUCN. So a total of 25% of all mammals are not assessed for their conservation status, which I think is something that of a priority for conservation measures. So there's also all sorts of collaborations between museums and different groups and organizations globally to do bat research um, more broadly, including many of the groups associated with GBATnet here. I've just included a snapshot of some of them. They're doing really great work and building capacity and or research capacity and training in countries globally, which is really important for doing all sorts of museum-based and conservation work, both in the museum and in the field. There are still some hurdles, though, with maintaining and growing museum collections, specifically with bats as it pertains to recent issues with COVID-19 and new regulations associated with it, and making sure that we don't accidentally spread disease to humans or spread from humans to bat populations. But some of the other major issues with trying to maintain museums is securing funding on a global scale. Um, there's a lot less taxonomists and systematists in certain groups or certain taxa. Other groups, there's a lot more now. But if we're not training the next generation to find this funding and build these collections, the museum world will eventually die, and we won't have this amazing resource for future research. Additionally, there's also public perception that we have to battle with consistently. People don't like seeing animals dead. Like we don't want to have to sacrifice these animals, but there's also important reasons that I've discussed throughout this talk that I think support the use of sacrificing animals for uh, broader research. Then, of course, there's international politics for collecting trips that can make it difficult to build capacity in regions where there should be more research done on these groups of living thing. So, with bats in particular, there's certain countries that there's been a lot of research done on bats, and others that there's been very little for various reasons. And then, additionally, I mentioned global inequalities. inequities here. I mean more so what I was talking about earlier in the talk with inequities in where museum specimens are stored versus where that research is actually being conducted. So, for the Global South, a lot of the history of the Global South has been colonial, and trying to move away from that, I think, is going to be, it's already happening, but it's still a priority for museum work in general. Just wanted to quickly thank some of my collaborators in various walks of my research, my advisor, Joe Cook and the Cook Lab, and anyone who's helped me in general with getting some of this work done, and all the museum curators out there doing God's work. Anyways, any questions? And thank you for letting me talk to you about museums for like 45-ish minutes.
Thank you so much, Connor. And yeah, we definitely have some time for questions. So I'll leave it open. You can put your questions in the chat if you like, or if you want to chime in, please feel free. Mhm. Oh, maybe something from Nancy?
Yeah, maybe we could get rid of the slides so we can see each other on screen. Anyway,
Good
there
point.
we go. That's nice. Great talk, Connor. Thanks a lot. I'm curious about your thoughts on sort of I guess, the future of museums, smaller museums at universities and things like that. There's been a trend over many years, I think, of universities deciding that museums are too expensive to support, that they take up too much footprint and things like that. What are your thoughts about that? I mean,
It's it's unfortunate to me that that is a huge hurdle in building museums, especially for people who want to build these museum collections in smaller universities or in areas where there isn't already a large museum present. Like the Museum of Southwestern Biology is huge for the region, so they already they branch out really heavily. But I know that there are a number of museum collections that have donated the full collection to larger museums, such as the Museum of Southwestern Biology, because there was no support to maintain the collection after the curator died. The Roush collection is one of those in Alaska. Ooh, I think he was actually in Oregon. I'm not going to state where he's from. I forgot actually. But that was a huge parasite mammal bird collection, and it all got put into the Museum of Southwestern Biology. Um, Same with a collection by I think Troy Best in Alabama. There was an Alabama collection. I think it was Troy Best. But unfortunately, if there is no institutional support for those museums, it's really hard to maintain it. Um, but finding external funding is probably going to be the best bet for maintaining those smaller ones. But again, we have to keep training museum scientists to go and build those collections or take over those small collections. Um, they are important. Like it's not, it shouldn't just be large collections monopolizing um, museum research. I, it's all there's already other major museums out there that have really low funding, even though they're huge, and really low staffing. And as a result, they can't get out loans to people. So people have to physically go there. It creates more cost and effort for the researchers themselves, yada, yada, yada. And that's often not a fault of the curators, that's a fault of the funding resources that are available to those curators, which is unfortunate. I don't know if I answered your question, but I can go off on that for like an hour.
oh
So,
no, that's good. I mean, I think that the being old, basically. I remember back when nobody got DNA out of museum specimens. Nobody did anything with pathogens in museum specimens. I think we're much more relevant to the rest of the world than we used to be. museum collections in general. But yeah, I mean, the next generation of researchers like yourself have to, carry the torch forward to support museum collections and the research that's based on them. So anyway, really liked your talk. Thank you.
To
Thank
kind of and hopefully I don't take too much of your pedestal, Connor, but to kind of build on both what you were saying and what Nancy was pointing out as well. I know I can speak personally. Some of my very first exposure to getting into the field of mammalogy, which then led me to researching bats, was through our small university mammal collection. And I went to a very small state institution where we didn't have access to as much research funding. So some of the very first exposure to working with wildlife was through museums. And even though we just had our small, like very small, tiny university mammal collection, we were still able to do small research projects that looked back into the history and population demographics of our local mammal population. So those small university collections are critical for helping new young researchers. I'm one of them. To get exposed to working with wildlife and understanding how museums can assist with different types of research. Yeah.
That's the perfect example. I similar when I was in my undergrad, I got taught mammalogy by an ornithologist. And so I didn't have a mammalogist there, but we had a little tiny collection of mammals, and I went through the entire thing by myself, reorganized certain things, updated taxonomy because nobody else was doing it. But it influenced a lot of my trajectory in my career because I really love that stuff. And I hope other people are also passionate about building these museum collections because it is, I think, basal to a lot of the research that is being done globally.
Yeah, I agree. I agree. Do we have any more questions for Connor? Okay. Well, then we can go ahead and wrap up our webinar today. Thank you everyone for tuning in to this month's iteration of My Field for Dummies. So please go and continue spreading the word about the importance of what museums do for not just bat research, but research in the wildlife sciences in general. The museums and museum repositories, big and small, are essential for the research and work that we do. So I don't think that I need to convince anyone too much in this room, but it's just another iteration of the importance of museums. And thank you, Connor, for the work that you do for helping spread the love of museums and museum collections and for coming to talk with us today.
Thank you. I really enjoyed it. Talk about museums anytime.
Thanks, Connor. And thank you, everyone, for joining us today. All right, and I will stop recording.